where these messenger RNA Frankenshots come from. DARPA awards Moderna Therapeutics a grant for up to $25 million to develop messenger RNA therapeutics. So notice DARPA is the Pentagon. Let me repeat that. DARPA is the Pentagon. The Pentagon bought, paid for, envisioned these mRNA Frankenshots. And I should also point out, Fort Detrick was involved in the development of COVID-19, the offensive biological warfare weapon at the University of North Carolina, BSL-3. So the Pentagon is both sides of the argument here. They're developing the weapon and the alleged vaccine.
The NIH and NIAID categorically has not funded gain-of-function research to be conducted in the Wuhan Institute of Virology.
He's going to say it wasn't gain of function. Many other scientists are already and have been saying for almost a year now that it all was gain of function.
In 2014, the NIAID initiated research project R01 AI110964, Understanding the Risk of Bat Coronavirus Emergence. As part of that project, over $3 million was sent to EcoHealth Alliance, and over $500,000 of that was sent to the Wuhan Institute of Virology. The requested work included this paragraph that many of our scientific sources told us was gain-of-function research. In 2017, EcoHealth Alliance's Peter Daszak and the Wuhan lab's Shi Zhengli published this progress report, which lists the ID R01 AI110964 in its funding section. This is the Fauci project. They analyzed bat coronaviruses and sequenced their genetic information. They then built various chimeras, genetically engineered new viruses, man-made in the lab. They infected human cells with them in the lab, and they then showed that their man-made viruses could replicate as a functional virus. NIH officials, Dr. Collins and Dr. Fauci, now claim that none of that is gain of function. Really? Here's Dr. Fauci speaking at a gain-of-function conference in 2012.
If you look at basic research as we've approached it, integral to that study has always been the issue of gain-of-function research.
He describes in detail the specific forms that gain-of-function can take.
There are a few ways to look at gain-of-function research. There's the naturally occurring mutations which naturally give gain of function, and investigators study these effects on the phenotypes of interest. Does this mutation make something more transmissible, more pathogenic, or adapt to host better? Or, what historically investigators have done is to actually create gain of function by making mutations, passage adaptation, or other newer genetic techniques such as reverse genetics and genetic reassortment. The NIH and NIAID categorically has not funded gain-of-function research to be conducted in the Wuhan Institute of Virology.
There is no question that there will be a challenge to the coming administration in the arena of infectious diseases, both chronic infectious diseases in the sense of already ongoing disease, and we have certainly a large burden of that, but also there will be a surprise outbreak. And I hope by the end of my relatively short presentation, you will understand why history, the history of the last 32 years that I've been the director of NIAID, will tell the next administration that there's no doubt in anyone's mind that they will be faced with the challenges that their predecessors were faced with.